Good morning. Hello everyone. I'm going to present my project on bioinformatics analysis of gene expression in healthy control stage 1, stage 2 and chemotherapy patient. Cancer is one of the leading cause of death worldwide. It develops when normal cells lose control over their growth and begin dividing uncontrolled. Understanding the change in the gene expression during cancer progression can help researchers identify biomarkers for early diagnosis and discover new treatment targets. In this project, I used bioinformatics tools to analyze gene expression data obtained from the NCBI Geo database. The dataset contains samples from healthy individual, early stage cancer patient, advanced stage cancer patient, and patient receiving chemotherapy. The analysis was performed using Geo2R to identify differentially expressed genes. After identifying significant genes, I used several bioinformatics tools such as volcanoplot, heat map, gene ontology, KEGG pathway analysis, string and cytoskip to understand the biological function pathways and interaction of these genes. The main objective of the project is to identify genes that change during cancer progression and evaluate how chemotherapy may influence gene expression. These findings may contribute to a better understanding of cancer biology and support the development of improved diagnostic markers and therapeutic strategies. Now, I will begin by explaining what cancer is. Before discussing gene expression analysis, it is important to understand what cancer is. Cancer is a disease which abnormal cells grow and divide uncontrolled. Normally, our body follow an organized process where cells grow, divide, perform their functions, and die when they become old or damaged. This process keeps our body healthy. However, in cancer, this normal control is lost. The abnormal cells continue to divide without stopping. As these cells increase in number, they form a mass of tissue called a tumor, and expectation is blood cancer, such as leukemia, where cancer cells circulate in the blood instead of forming a solid tumor. Cancer cells also have the ability to invade nearby tissue and spread the other parts of the body through the blood of lymphatic system. This process is called metastasis, and it is one of the main it is one of the main reason cancer become life threatening. Cancer develop because of changes or mutation in genes that control cell growth and divisions. These mutation may occur due to inherent genetic factors, smoking, environment, pollutions, radiations, infection, or unhealthy lifestyle habits. In my project, I study gene expression changes associated with cancer by comparing healthy individual cancer patients at different stage and chemotherapy patients. We can identify genes that are involved in cancer development, understand how chemotherapy affects gene activity. Now, I will explain the difference between a normal cell and a cancer cell. A normal cells grow, divide, and die in a controlled manner. This process is called the cell cycle. When a cell becomes old or damaged, it undergoes apoptosis, which is programmed cell death. This helps maintain healthy tissue and organs. It contrasts a cancer cell lost this control instead of stopping growth. It continues to divide rapidly and uncontrolled. Cancer cells ignore the signals that tell them when to stop dividing or when to die. Another important difference is that normal cells stay in their original location and perform a specific function. Cancer cells invade nearby tissue and spread to other parts of the body through the blood or lymphatic system. This process is called metastasis. Normal cells have a regular shape and organized structure, while cancer cells often have irregular shapes and abnormal nuclei. They also consume more nutrition and energy because they divide very quickly. In bioinformatics, we compare genes expression between normal cells and cancer cells. This helps us identify genes that are either upregulated or downregulated. Upregulated means more active, and downregulated means less active in cancer. These genes can be used as biomarker or potential target for treatment. Structure of normal and cancers. Now, I will explain the structural difference between normal cells and cancerous cells. This image on the left representation normal cells, while the image of right represents cancer cells. But let's look the cytoplasm. In normal cells, the cytoplasm is large and provide enough space for normal cellular function. In cancer cells, the cytoplasm becomes smaller and nuclei occupies more space. Next is the nuclei, which is the control center of the cells and contain the genetic material or DNA. Normal cells usually have a single nuclei, whereas cancer cells may have multiple nuclei due to uncontrolled cell division. Inside the nucleus is the nucleus, which is responsible for producing ribosome for protein synthesis. Normal cells have a single small nucleus, which cancer cells have multiple and enlarged nuclei because they need to produce more protein to support rapid growth. Another important feature is chromatin, which contains DNA. In normal cells, chromatin is fine and evenly disturbed in cancer cells. Chromatin appears coarse and dense because of normal DNA organization and genetic mutation. Overall, normal cells have regular shape, control growth, and normal cellular structure. In contrast, cancer cells have a irregular shape, multiple nuclei, enlarged nuclei, coarse chromatin, and divide uncontrolled. These structural changes are important indicators of cancer and help scientists and doctors distinguish cancer cells from normal cells. They are also associated with changes in gene expression, which is the main focus of my bioinformatics analysis. Now, I will explain how cancer cells develop. Our body naturally produces new cells every day to replace old or damaged cells. Normally, the immune system recognizes abnormal cells and destroy them before they can cause any harm. However, sometimes a few abnormal cells escape detection by the immune system. These cells survive and continue to divide uncontrolled. Over time, they multiply and form a mass of abnormal cells called a tumor. As cancer cells develop, they become very different and healthy cells. They no longer follow the body's normal signals that control when a cell should grow, divide, or die. Instead, they continue growing even when they are not needed. Cancer cells also lose their normal functions. They do not communicate properly. When neighboring cells are ignored signals and normally stop cell growth. This allows them to multiply rapidly. As they continue to grow, cancer cells acquire additional genetic changes, making them even more aggressive. They can invade nearby tissue and eventually spread to other parts of the body through the blood or lymphatic system. This process known as metastasis. In summary, cancer cells develop when abnormal cells escape the immune system, accumulate genetic mutations, lose normal growth control, and continue dividing without stopping. These changes in cell behavior are closely associated with changes in gene expression, which is the focus of my bioinformatics study. Now, I will explain how DNA damage and can lead to the development of cancer. This figure shows the difference between a healthy cells or disease or cancer cells. At the top of left, we can see exogenous damage. Exogenous damage means damage caused by external factors such as cigarette smoke, air pollution, radiations, harmful chemicals, and ultraviolet rays. These factors can damage and DNA inside our cells. Our body has a natural DNA repair system. When DNA is damaged, repair proteins detect the damage and fix it. If the repair is successful, then cells continue to function normally. As shown on the right side, in a healthy cell, the rate of DNA repair is greater than the rate of DNA damage. This means the cells can repair most DNA damage and remain healthy. Every day, a normal cells experience thousands of DNA modifications, but efficient repair mechanisms maintain normal cell function. However, sometimes the DNA damage is served or repair system fails. The DNA damage remains unrepaired, leading to permanent mutation. These mutations can affect both nuclear DNA and mitochondrial DNA, causing abnormal cell behavior. When DNA damage is not required, the cell may enter pathology, which means the cell becomes abnormal and diseased. At this stage, several outcomes are possible. The cell may undergo apoptosis, which is programmed cell death, and senescence, where all the cells stop dividing permanently. If this protective mechanism fails, the damaged cells continue to divide uncontrolled and eventually develop into cancer. Therefore, in a disease cells, the rate of DNA damage becomes greater than the rate of DNA repair. As a result, mutation accumulate normal cells, regulations is lost, and cancer begin to develop. This process explains why maintaining DNA repair mechanism is essential for preventing cancer. In my bioinformatics project, I analyzed change in genes expression to identify genes involved in DNA damage, DNA repair, and cancer development. These genes may serve a potential biomarker or therapeutic target for lung cancer. What is chemotherapy? Chemotherapy is one of the most common treatments used to treat cancer. It uses anti-cancerous drugs to destroy cancer cells to stop them and from growing and dividing. Cancer cells divide much faster than normal cells. Chemotherapy targets these rapidly dividing cells by damaging their DNA or blocking the process of cell division. As a result, cancer cells cannot multiply and many of them eventually die. Chemotherapy drug travel through the bloodstream, allowing them to reach cancer cells throughout the body. This makes chemotherapy especially useful for treating cancers and have spread beyond their original location. Chemotherapy cannot always distinguish between cancer cells or some healthy cells. They also divide rapidly. These include cells in the bone marrow, hair follicles, digestive tract, and rep- reproductive system. Because these healthy cells are also affected, patients may experience side effects such as hair loss and vomiting, fatigue, mouth sores, loss of appetite. Now, I will explain how chemotherapy works. This diagram shows the step-by-step process of chemotherapy treatment. First, cancer cells grow and divide uncontrolled, forming a tumor. Second, chemotherapy drugs are administered and enter the bloodstream. Third, these drugs travel throughout the body and reach the cancer cells. Fourth, the drugs damage and DNA or stop cell division, preventing cancer cells from multiplying. Finally, the damaged cells, cancer cells die and the body immune system gradually remove them. Chemotherapy mainly targets rapidly dividing cancer cells. It can also affect some healthy cells that divide quickly, such as those in their hair follicles, bone marrow, and digestive tract. This is why patient may experience side effects like hair loss, fatigue, and vomiting. Overall, chemotherapy helps control cancer growth by stopping cancer cells from growing and spreading. Common side effect. I will be taking about some of the most common side effects of chemotherapy. The first side effect is fatigue. Many patients feel extremely tired even after getting enough rest. This happens because chemotherapy affects the body energy level and healthy blood cells. Second, common side effect is nausea. Some people may experience as upset stomach or vomiting during or after treatment. Doctors prescribe anti-nausea medications to help manage these symptoms. Another important side effect is immune suppression. Chemotherapy can reduce the number of white blood cells, making it harder for the body to fight infections. Patients are often advised to practice good hygiene and avoid contact with people who are sick. Finally, hair loss is a well-known side effect. Since chemotherapy targets rapidly growing cells, it can also affect hair follicles, causing temporary hair loss. In most cases, hair begins to grow back after treatment ends. Conclusion. While chemotherapy can cause challenging side effects may Many of them can be managed with the proper medical care and support. Understanding these effects help patients and their families prepare for treatment and cope with the journey more confident. I will explain the bioinformatics workflow used to identify important genes and biological pathways associated with a disease. Step 1. Geodatabase. The workflow begins by selecting a suitable gene expression dataset from a gene expression omnibus geo database. This database contains publicly available gene expression data from various biological studies. Step 2. Geo2R. Next, we use Geo2R, an online analysis tool that compares two groups such as disease samples and healthy control to identify difference in gene expression. Step 3. Differentially expressed genes, DEGs. These compare, we identify differential expression genes or DGs. These are genes that significantly unregulated and downregulate between the two groups. Volcano plot. The DGs are the visualized using a volcano plot. This graph helps us quickly identify genes that are both statically significant and healthy differentially expressed. Heat map is created to display an expression patterns of the selected genes across all samples. This allows us to observe clustering and difference between the groups. Gene ontology, geoanalysis. The identified genes are analyzed using gene ontology for go analysis to determine biological process, molecular function, and cellular components in which they are involved. KEGG pathway. KGG pathway is performed to identify the signaling pathway and biological mechanisms that are significantly associated with the differentially expressed gene. String database is a protein-protein interaction network showing how the protein interact with one another. Cytoscape. This interaction work network is imported into cytoscape where it is visualized and analyzed to identify hub genes, which are highly connected genes that may play critical role in disease progression. Stop. Step 10. Biological interpretation. The results from all analysis are interregulated to draw biological conclusion, identify potential biomarker, and suggest possible therapeutic target for future research. Conclusion: the bioinformatics workflow systematically move from raw genes expression data to meaningful biological insights. It helps researchers identify key genes, understand disease mechanisms, and discover potential targets for diagnostic and treatment. Difference gene expression analysis of GES10072. GES10072 is publicly available dataset from the NCBI Expression Omnibus database. It contains 58 samples, including 49 cancer samples and 9 normal tissue samples. The analysis was performed using GEO2R tools, which is based on Lemma statistical package. In this video, I will show you how to search for open a gene expression dataset on NCBI Geo database, the gene expression omnibus. We all be looking at a well-known lung cancer dataset called GSE10072, which contains gene expression data from 58 lung tumor samples and 49 normal lung tissue samples. First, open your web browser and go to the NCBI Geo homepage. You can either search NCBI Geo on Google or direct it to this address. Once you are Geo homepage, you will see a search box at the top. This is where you will type in the accession number of the dataset we are looking for in our case. After searching, you will land on Geo accession display page for CSE10072. This page gives all the important details, the title of the study, and the summary of experiment, the number of samples, the platform used, and links to download the data. If you scroll down, you will find a table listing every individual sample in the dataset, each with the GSM accession number. Clicking on any of these will show you the sample specific detail, like whether it's a tumor or a normal sample. Now I actually download. Now I prepare the normal cancer a case control and the lung cancer is the cases. This page, this page gives you all important information. Now we prepare the control. We select all the control and the cases. So we have total 14 cases. So we have 9 samples and the normal lung tissue we call control. So we select all the control. Now we select all the lung tumor and the normal samples. So we have total 15 samples and 11 control. Now we analyze this. The data will be processing. Now I actually download the data for analysis. Scroll to the bottom where you will see the download family sections. Here you have a few options. The series matrix file is the easiest one to use, especially if you are looking R or Python. So the simple recap, go to the Geo website, search using the accession number, open the data page to see the summary and sample and download the series matrix file to start your analysis. That's the complete process of finding and opening a cancer gene expression dataset. This is the result. Next, I use Jupyter. Try in your browser, then Jupyter Lab. Click the Jupyter lab. Now, create a new notebook. In the top right corner, click the new drop-down menu. Select Python. A blank notebook will open in new tab. Click on the next. Let's say Untitled at the top of the page. Type new name and click Rename. Write and execute content. Choose to write and run Python script into your first cell and press Shift plus Enter to execute it. The output will appear directly below the cell. Use to add formatted text, notes, and title. Select Markdown from the toolbar. Use hashtag for a header title. Write your text and press Shift plus Enter to render it correctly. This slide shows the volcano plot of differential express gene identified from GSE10072. The x-axis represents log fold changes which show whether genes are upregulated or downregulated. Genes on the right are upregulated while those on the left are downregulated. The y-axis represents the negative log 10 of the p-value, which indicates the statistical significance of each gene. The higher a point appears on the graph, the more statistically significant on the gene is. In this plot, red dots represent upregulated genes, blue dots represent downregulated genes. Overall, the volcano plots provide a quick visual summary of the gene that shows the largest of the most significant expression genes between cancer and normal cancer. These significant genes are selected for further analysis including heat map visualization, CO and KEGG pathway enrichment, and protein-protein interaction network analysis. Upregulated genes. The top upregulated genes is SPP1, SP1, NK1, COL11AI, MMP1, TOX3, MMP12, SULF1, COL, A1, GREM1. In this plot, red dots represent significantly upregulated dots, blue dots represent significantly downregulated genes, and gray dots represent genes that are not significantly differently expressed. This slide shows the histogram of the p-value obtained from the differential gene expression analysis. The x-axis represents the p-values ranging from 0 to 1, while the y-axis represents the number of genes. As we can see, most genes are concentrated near p-value close to 0, indicating that many genes show statistically significant differences in expression between the cancer and normal tissue. In contrast, genes are higher p-value and less likely to significant differential expressed overall. The histogram confirms that the dataset contains a sustainable number of significantly differential expressed genes, supporting a reliability of the subsequent analysis such as volcanoplate, heat map, and pathway enrichment studies. This slide shows the list of differentially expressed genes identified from the GSE110072 dataset after GO2R analysis. Each row represents a gene along with important statistical value such as the adjusted p-value for genes, and the gene symbol. These genes are ranked according to their statistical significance. We selected genes with adjusted p-value less than 0.05 and an absolute log fold changes greater than or equal to 1, ensuring that only genes are significant expression changes were included. From the table, ser- several genes such as CD36, AGER, and all shows differently downregulated cancer tissue compared to normal tissue. These differentially expressed genes form the basis of next step of the study, including functional enrichment analysis, pathway analysis, and indication of key hub genes associated with cancer. This is present the GU2R differential genes expression result obtained from the GSE11072 dataset. The table includes probe, IDS, adjust p-value, raw value, moderate statics, p-statics, log fold changes, and gene symbol, and gene description. Genes with adjust p-value less than 0.05 and greater than equal to 1 were considered significantly differential expressed. In this dataset, genes such as CD36, AGR, ANDRD, CA4, HIGD, 1B, and RAMP2 show significant downregulation in cancer samples compared with normal tissue. In conclusion, this project highlights how bioinformatics can be used to identify important genes, biological pathways, and molecular mechanisms associated with chemotherapy and cancer treatment. These analyses provide valuable insight that may support discovery of new biomarkers and the development of more effective and personalized therapeutic strategies. Thank you all for your time and attention. I hope this presentation has given you a better understanding of the role of bioinformatics in chemotherapy research. I would be happy to answer any questions you may have. Thank you.